All right, so we left here at our different transmembrane proteins, and these are all proteins that exist within the plasma membrane. So here we see extracellular fluid out here, the inside that cytosol on this side here. So we've got our receptor proteins. So these proteins are going to exist through the whole membrane, and we can get different concentrations of molecules outside of the cell, and those molecules can bond to receptors to tell the inside what to do. These could be hormones as well that will tell the cell to start producing some sort of chemical or start processing something else. But we've got these receptor proteins that are going to regulate what the inside of the cell is doing. We've got recognition proteins. Okay. These exist. They're mostly carbohydrate chains like we see up here that will tell other cells. A lot of times this will be things like white blood cells that these are our cells. Don't attack these. They're safe. We also have transport proteins, which will allow certain molecules into and out of the cells. We've got what are called aquaporins to allow water into and out of the cell. And then there are a lot of other of these transport proteins for other specific molecules to allow them in and out. Enzymatic proteins, these are enzymes that exist that will help speed up reactions on this plasma membrane. Okay. So now we're going to look at the actual movement through this plasma membrane. So what are the different ways that molecules can go from one side of the cell to the other side? Okay. So the first one that we're going to look at is diffusion. Okay. So with diffusion, we really look at concentration gradients. Okay. So concentration, the number of molecules in a given unit of volume. So here we've got this drop of red dye, and it's concentrated in this area, not very concentrated over here where there are no dye molecules. So what we see is through the process of diffusion, molecules will move from an area of high concentration to low concentration. So you can do this at home. You can drop food coloring in water, and what you'll see is at first it'll stay in one little area. But then eventually those molecules will start to spread out from one another until they filled that entire liquid. Right? And so it finds this equal concentration, this equilibrium across the whole fluid. And so what we see is in our body, this will happen as well. Molecules will move from an area where there's a lot of that molecule, and they'll kind of spread themselves out. This is a type of passive transport. When we talk about passive transport, we're looking at the movement of molecules across the membrane that does not require energy. So what we see is that this is partly due to molecular size. Things that are really small can usually move through this membrane without a lot of problem. Lipid solubility, remember those phospholipids are lipids. So if something is soluble in lipids, then it has a much easier time moving through that phospholipid bilayer. And then also the size of the concentration gradient. If we're already pretty close to equilibrium, we're not going to see a lot of that movement. But if there's a clear gradient of very high concentration on one side of the membrane, low concentration on the other, we're going to see some of that movement, not requiring energy. So we've got our simple diffusion, which is movement from high to low concentration. We've got facilitated diffusion and osmosis as well, which we'll talk about on these next few slides. So simple diffusion, again, just movement down the concentration gradient from high concentration to low. So we see this here. We've got our molecules of dye over on this side of the membrane, and they start moving to the other side until they reach equilibrium. Now this can happen with different types of molecules as well, right? Purple's going to move to the right while green moves to the left, and eventually we reach this point of equilibrium. Okay? They move down their concentration gradients for that specific type of molecule. Now, it will get more complex than this, but we're not going to worry about ion concentration and things like that. Things that do this: oxygen, carbon dioxide. Okay, these are very small molecules that have a pretty easy time moving directly through that membrane because they're so small. Also, lots of lipid-soluble vitamins can make it through. And how about water? Well, the diffusion of water is a special type. Okay, it's got its own name, which we'll talk about. So the phospholipids, again, things that could move through: oxygen, carbon dioxide, lipid-soluble molecules. Okay, so things that can move through: small, nonpolar molecules. Okay, and they have an easy time going directly through. So let's take a look at facilitated diffusion. Okay, again, because it's diffusion, it does not require energy. Here we're looking at water-soluble molecules in and out of the cell. So these are things that will not move directly through the membrane because they're not lipid-soluble. So what makes facilitated diffusion different is that here we're using a protein, right? Which is why it's facilitated. It needs some help. So we get this protein. In this case, it's a channel that these molecules are going to move from high concentration to low concentration through that protein. Okay, we have this with glucose, various amino acids, and so this is facilitated diffusion. Okay, molecules move down the concentration gradient from high to low with the help of a protein. Okay, and now our third type here is osmosis, and so this is just diffusion of water. So we're moving from an area of high concentration of water to a low concentration of water through that membrane. The other way we will sometimes see this written is that it'll move from an area of low concentration of solutes to high concentration of solutes. Oftentimes within our cells, water can move quite easily, but these other solutes that we have to equalize these concentrations of can't make it across the membrane. So it's water that's doing a lot of this movement. Okay. So the concentration of water depends on what's dissolved in it. We're looking at the strength of that solution. So the solute is what's being dissolved. So here we've got salt being dissolved in the water, and the solvent is the thing doing this dissolving. Most of the time, this is water. Okay. Lots of solutes in cells, right? We rely on lots of different molecules for various different macromolecules, which will bond together and all of this. And we've got lots of ions that will help carry out various functions. So we've got this cytosol that's just full of all of these solutes. So to equalize these concentrations outside and inside of the cell, water is going to move across this plasma membrane in order to balance this out. And we've got a couple of ways that we can do this. Water can move directly through the plasma membrane because it's a really small molecule. But we've also got these aquaporins, these channels that will help water move more easily into and out of the cell. Okay. So this is based on tonicity. The word tonicity refers to the concentration of those extracellular fluids. Okay, so we've got a couple of different terms that we'll look at here. So we've got isotonic. Iso means same. So here the concentration outside of the cell is the same as inside of the cell. So water is still going to move in and out of these cells that are isotonic, but we're not going to get an overall shift. It's going to be the same moving in as the same moving out. And so we see that this is the goal, is to get to be at this isotonic solution. Okay, so we will hit our hypertonic and hypotonic in this next video.